realized that this is me between the lunch and the talk. And actually, I found it pretty amazing. I almost forgot how well AET feeds the attendees. And there seems to be constant flow of food every day and keep you going on. And this is actually what we need in our discovery work. That we need a lot of samples to feed our discovery flow, and our company really is a company focusing on the discovery on antibody discovery from lead generation to cell line development. And the word here is kind of small, but we are really focusing everything between cell line development, especially the hits generation and the hits characterization area. And I will tell you more about our discovery service, but everything needs to be supported by a lot of sample generation for your testing and keep the project going as fast as you can. And this is all supported by the high-throughput antibody production platform that our company has. I think Biointron really, our service can really be divided into three major parts. And the Biointron Express is the major part of that. And we also have a Biointron Discovery, which I hope to tell you a little bit more later in the talk for a few minutes. And our catalog service really provides bulk, cost-effective reference antibodies, biosimilars, and anti-mouse target antibodies that supports a lot of academic research and as well as in vivo efficacy studies. So the next few slides, I'm going to tell you more about the expressing service that we are specialized in. And this talk really is intended to be introductory and tell you what we do. And basically, I hope to have a chance to tell you more about in depth. But basically, the take-home message from us is we have the capability and the capacity to do high-throughput antibody production in 1 to 2 weeks for any type, any species. And from the moment that we receive your sequence, and we will start our own in-house gene synthesis process and transfect it into the host cells such as the CHO and the HEC293. And we can do this for not only simple mAb, but also complicated bispecifics, multispecifics, in the scale of a few hundred micrograms to a few hundred grams in scale. Not only that, we routinely receive orders from dozens to more than 1,000 at the same time, and we deliver those samples in 1 to 2 weeks as well. Then how can we achieve this? We really, when I actually worked as a researcher many years ago, we are always bothered by how long the process takes to generate antibody samples. And as I remember, it usually takes about 6 to 8 weeks at a time to get the protein antibody samples. And I have to outsource the gene synthesis and do in-house plasmid extraction and do transfection, purification. And our process really, our company really streamlined the whole process and let it become one streamlined flow from in-house gene synthesis to pipetting, even to labeling, aliquoting, everything. So a lot of automation and the process optimization was done to shorten the timeline to make possible. And I can confidently say that we make the fastest delivery in sample preparation in the industry at this scale. Looking at actually how the scale and the capacity that we have, we actually make more than 50,000 maps every year. I mean, I think this year, this number is more than that. 50,000 in various scales. And most of that, this is just a map-like format. And I categorize this by, the majority of them is still regular IgG maps, but there's a pretty significant amount of VHH, FC, followed by, VHH with a tag, as well as some single-chain FVs. And in terms of the species and the subtypes, the majority is still IgG1, followed by IgG4, with some mouse antibodies as well. And whatever types of antibodies you request, we have historically, from the beginning that we start to provide the service, historically we have expression levels in the hundreds of milligrams per ml in a transit transfection. And the average, as you can see that for single-chain FVs or for his tag proteins, the yield might be a little bit lower, and especially for single-chain FVs, the purity could be a little bit lower too, which is commonly acknowledged in the industry as well. But we strive to make it more, make it better. And we also see that we have, over the years, we accumulate the data for the expression titer for human, mouse, and the rabbit. And again, the human IgGs are probably the best because not only we receive more orders, but we spend a lot of time to optimize it. And I tell you the continuous optimization of that. In terms of purity, you can see that we maintain a very high purity after one-step purification, mostly in the mid-90s, in the high 90s, meeting customer request, QC request. And this is an example. Because of our capacity, we can handle a lot of samples at the same time. This is an example from one company working on AI designs. And it's a while ago. The client asked us to express a panel of 818 antibodies with our HTP service. And we, from text to test, meaning that from the moment that we receive the sequence to the moment we ship out the samples, purified samples, and they received, we spend 10 days in one single batch. And that way, they can quickly get into the assays, quickly verify how their design goes, and iterate more cycles for the and the quality of that. And I did a little bit stats here, and you can say that you can tell that the majority of them are between 100 to more than 300 mg per liter in terms of titer. But this is a very small scale. And more than 95, the vast majority of them had a one-step purification, reaching more than 95% purity. So this is already pretty good, right? But we strive to make it even better. So we developed a rapid map production in a 4-day culture format. And this is telling you, some of the examples here. So in another example of about 150 maps, in 4-day culture, we optimized the process, and then we can reach on average of 322 mg per ml titer, and with a 97% average purity for all of them. So this is just 2 examples for map. We got a lot of requests for VHH these days, and the most common format, as I mentioned before, was a VHH FC fusion production. And this, in the early days, this is a one batch of about 180 molecules made in made in a 10 ml culture format. And you can see that the average delivery amount is about 2 mg for each of those samples. But this is not the end. In recent days, we also try to shorten the time and by optimizing the culture conditions and optimizing the process. And in this example of about 60 molecules using 4-day single batch format, we were able to deliver more than 5 mg product for each of the sample on average. There's also a constant need of making the whole process more efficient and cost-effective. And instead of the normal 10 ml, 30 ml cultures that we do, we also developed, plate-based 24-well and 96-well high-throughput expression in 4 days. And this provides, even faster sample production and probably more efficient storage situation. Because just imagine 1,000 sample requests from an AI company. They got a mix of samples, and they probably just test once. And how are you going to store them? That's a very practical question. And in this case, using in a 24-well format, we deliver on average. This is a single chain VFC format. You can see on the left. Intrinsically, we found the expression level could be a little bit lower, but we could still reach 100 microgram per ml titer, which is enough for most of the customers. We also have extensive experience in bispecific formats, and we actually, this year, we reached more than 40,000 molecules this year, and the number is still growing. And we are familiar with all these commonly seen formats. And I'll tell you a little bit more about these common formats and their expression. In this case, the knobbing hole format was divided into 2 types. One is using common light chain. One is using each individual pairing. And you can see that the majority of that still requires with a different light chain. But in terms of expression, you get a better titer when the format use common light chain. And the purity, however, from one step is pretty consistent these days. And we do have, in this one-step purification, we could reach more than 95% by SDS, as well as SEC. This is atypical because I think knobbing hole is pretty common these days, and we have a lot of experience work on that. And there are occasionally cases that requires 2-step purification in this format. And in this case, after 2 steps, we reached, we could improve the purity from 89% to more than 99% with a yield of 49% here. Not only that, IgG, VHH these days are extremely popular, and you can see all kinds of formats, variants that people ask us to do, symmetrical or asymmetrical with, the renditions are limitless. And I just tell you a little bit about our experience here. So by stats, we still see that the VHH connected to the carboxy terminal of the heavy chain is still the most commonly, most popular format, and followed by the N-terminal fusion. And in terms of titer produced, the heavy chain C-terminal fusion give pretty good expression level, and I think it's among the best of that. The purity, however, it's pretty similar. It's probably still the best among the 4 basic formats that we commonly see. The worst one is probably the light chain C-terminal fusion. And I just give you some one-step purification example here, nothing special here. We still see a lot of requests of conventional IgG fused with a single-chain FV. This also can be divided into some basic formats, as well as some fancy extended variants. And we again did the stats on that. You can see that the heavy chain C-terminal fusion is still the dominant format people prefer. And indeed, the expression level is second, not the best. Interestingly, the light chain carboxyl terminal fusion probably give you the best expression titer. But the number is much smaller, so statistically, the significance could be a little bit questionable. But we see that the purity of these bispecifics is visibly lower compared to the VHH fusions. I think that this is also quite understandable. The single-chain FVs are known to be pretty, picky, and you generally after one-step purification, you see 80% or even 70% on average purity. That requires probably second-step purification. These are just some one-step purification examples for you to see. Yeah, and so I tell you about these antibody expression platforms, high throughput, high yield, very efficient, high capacity. But how we can, but really this platform provides a very powerful tool for us to do other things, such as, I give you an example for the affinity maturation here. So the distinct features of our affinity maturation process is that we actually have a full coverage of all 6 CDRs with a non-biased mutagenesis for saturation mutagenesis, and there's no display system needed. And we use affinity ranking ELISA to identify the hotspots and apply. Because we have our own high throughput antibody production platform, we can do this in 6 weeks, and compared to using a library approach in months. So how we achieve this? This is really nothing new. Conceptually, this is very old. When I was working for Johnson & Johnson 20 years ago, I mean, we already know that MedImmune had a publication of finding those individual hotspots and combining them, making a library, finding the hotspots and combining the hotspots for secondary screening. But using a library system could cause bias because the display system itself has preference, and maybe in the bacterial system, this may not be friendly to your, may not reflect the true affinity situation that you got from panning. So what we do is, for each position, for each CDR position, we generate saturation mutations and pick one plate randomly. So in that way, 88 clones were picked. That's more than enough statistically to cover all 17 amino acid variants. And we make 70 plates for each position. In that way, every position is covered. And then we can use ranking ELISA, normalize the signal, and find out and discover the hotspots. Discover the hotspots if certain position, certain mutation can give us improvement in terms of affinity. And then combining these, in a secondary round, we combine these mutations, hotspots, and then went through the high throughput expression again, and use ELISA or facts to identify even better binders. This way, you can make sure all the data you got is equally represented, and there's no surprise there, and you don't miss anything. And I just give you some example of a classic mouse antibody humanized, and in this case, we were able to use the method to improve the affinity by more than by close to 120-fold. And one of the most interesting cases is you can use this to do interesting things. One client asked us to improve the cyno affinity, significantly improve the cyno affinity, but maintain the human antigen binding because the difference between the two is too big. And we were able to achieve that, improving the cyno binding by 50-fold, but maintain the same affinity against the human antigens. These days, again, VHH is very popular, and we get requests for VHH affinity maturation all the time, and we can do affinity maturation just specifically on K-on, and in this case, we were able to get 7-fold improvement, more than 8-fold improvement on the K-on, which is normally a little bit slightly harder in the format to improve K-on. K-off is no problem. I mean, K-off, you just wash more and let it sit more. And we can, in this case, we improve the affinity by more than 20-fold. So this is basically a very brief introduction of our high-throughput purification. And we have now, in China, we are very well known, but in the US, we are rapidly recognized by our clients, our customers, to have the ability to achieve the sample preparation in very short time with high quality. And not only that, expression is not the only thing that we do. We actually have a very strong platform for alpaca VHH discovery. And I will spend just 5 or 6 minutes on this. Yes, what we do is fully take advantage of the fact that we have two alpaca farms that we own, and we constantly keep close to 400 animals, and we acquired them when they were young, like 6 months. So we keep track of all the records, the health records, we make sure they are naive, and we raise them until they're ready for immunization. So because most of our clients are in the pharmaceutical industry, they prefer using naive animals to start with. And after immunization, we typically use a phage display as the most popular way to construct immunized library for panning. There's also ways to do single B cell sorting, which I hope to tell you more later. And some clients would just ask to do NGS sequencing, and they get big data to analyze. So the process is really not very difficult. I mean, I'm going to tell you one example using one T cell receptor antigen as an example, and this is a client project that we generate immunized libraries and pan the library both by solid and liquid phase. And after heat characterization by ELISA FACS and the functional assays, we identified the leads and went ahead, humanized the hits, and construct bispecific antibodies for the client. So quick review of the data here. And in this particular case, we identify 49 antibodies that bind to target X, shown here by SPR. The affinity range is between, mostly is in the -8 and the -9 range. And there's a couple of them even in the -10 to start with. And then 43 of them were able to bind against the overexpressing cell lines. And we went ahead to show that among them, 31 showed agonist activity compared to the positive control. And the blocking, ligand blocking assay was also performed to show that the majority have ligand blocking assays. This is just a quick example, but we often get a request that as multiple antigens can our customer would like to work on multiple antigens, and we can actually immunize the animal with the 4 antigens at the same time, and pan them separately against the target. And this one showed you that we collect the blood samples at a different stage whenever the titer comes up, so we capture all the diversity at the right time. And pan against the 4 targets, and you can see that in this case that we got pretty high good binders against all 4 targets. And this is a very efficient way to do immunization and screening. And after that, our affinity measurement, we have capacity to use a vehicle AK to do a normal affinity measurement, but as well as epitope binding by using Catera. We are probably we work closely with the Catera people to develop protocols for this in this area. So this is just give you one last slide to give you some idea about what kind of affinities that we get from our alpaca discovery projects. This is nothing scientific here. This is a stats that from 30 different projects, different targets from one client. And there's no scientific measurement here. We simply, we got thousands, more than 1,000 binders. And because more than 600 of them were put on SPR characterization, and we just simply look at what kind of affinity we can get. And you can see that the vast majority of them actually fall into the -8 and the -9 with pretty good numbers in the sub-nanomolar category. So the numbers are still accumulating, but I think we are probably the biggest company that serves the alpaca discovery, VHH discovery that I know. And we've been doing a lot of targets simultaneously as we speak. I don't think I have time to introduce the single B-cell sorting platform, but just simply say that we use a microfluidics platform to sort oil-based droplets, and then combining the NGS sequencing and our high-throughput expression to identify hits in 1 week and get the binders and the binding data in 3 weeks or 3 to 4 weeks after immunization. So this is a very efficient way and a quick way to get your binders using the single B-cell platform. So with that, I think I will skip the PD-1 case. Just tell you that this is an example of the case that we identified novel PD-1 hits. That you can see that in this particular case, we got a lot of epitope similar to Nivo, indicated by the orange sign here. This is the Nivo. And we found a lot of similar to that, but nothing nothing similar to Keytruda, which is a very interesting observation. Anyway, we have a booth here, 103. Well, you're welcome to visit us to learn more, and And our offices are located in New Jersey to make sure the shipments can reach you in time. From our production site in China, we are headquartered in Shanghai, and also have logistics centers in Europe, and to serve our overseas customers. And thank you very much for your attention. Thank you.